In this lecture, we will talk about carbohydrates. Carbohydrates are the essential part of the human diet, and excess carbohydrates can contribute to the development of the disease, such as diabetes. In this example, we can see that pre-meal number for a patient with diabetes should be 110 milligrams per deciliter or less, and if it goes up by more than 50 milligrams per deciliter, the amount of carbohydrates that is consumed by the patient should be lowered. Carbohydrates serve as one of the major, if not the major, source of energy from our diet. They are composed of carbon, hydrogen, and oxygen, and they're also called saccharides, meaning sugars. Many carbohydrates, such as glucose, are produced by the process of photosynthesis in plants, where energy of light is absorbed in chloroplasts, and that energy is used to convert carbon dioxide and water into carbohydrates. In living cells, carbohydrates are oxidized to carbon dioxide and water in the process of respiration. Essentially, the processes of photosynthesis and respiration are two opposite processes. Carbohydrates are generally divided into monosaccharides, which are the simplest carbohydrates, disaccharides that consist of two monosaccharides connected to each other by the covalent bond, or polysaccharides, which contain many monosaccharides in the long chains that are sometimes branched. Typical monosaccharides consist of 3 to 8 carbon chains, carbon atoms, with one carbon in the carbonyl group. If that carbonyl group is in the aldehyde, we call such monosaccharides aldoses. If this carbonyl group that is in the monosaccharide is in the keto group or ketone group, we call such monosaccharides ketoses. Monosaccharides have hydroxyl groups on all other carbons except the carbon that is the part of the carbonyl group, like this one. Depending on the number of carbons, monosaccharides can be classified as triose, tetrose, pentose, and hexose. For instance, an aldopentose is the monosaccharide that contains 5 carbons. One of them is an aldehyde group. Ketohexose is a saccharide that contains 6 carbons. One of them is in a keto or ketone group. Here are some examples. Glyceraldehyde is the aldotriose. Threose is aldotetrose, 4 carbons, aldehyde group. Ribose is aldopentose, 5 carbons, one of them is an aldehyde group, and fructose is a ketohexose, 6 carbons with one of them in the ketone group. Now we need to talk about the concept of chiral molecules. The concept of chirality refers to the fact that two objects, although being practically identical, cannot be superimposed on each other because they have mirror images. They have mirror symmetry, like your left and right hands. To understand the concept of chirality, we first need to understand the concept of structural isomers. Molecules are called structural isomers when their molecular formulas are the same, but bonding arrangements in those molecules are different. For example, ethanol and dimethyl ether are structural isomers. They have the same atoms, but they are arranged differently. Propanol and propanone are two structural isomers because they have the same number of carbons, hydrogens, and oxygens, but these carbons, hydrogens, and oxygens are arranged differently. Stereoisomers not only have identical molecular formulas, they are not considered to be structural isomers. In stereoisomers, atoms are bonded in the same sequence, but atoms differ in the way they are arranged spatially. For instance, when you have two organic molecules which are essentially the mirror images of each other, those mirror images cannot be completely matched, which means they are non-superimposable, and that means they are chiral. So essentially, chiral molecules have the same number of atoms that are arranged differently in space, and they represent two non-superimposable mirror images of each other. So going back to our hand analogy, hands are like chiral molecules. They are mirror images of each other and cannot be superimposed. When you have two molecules which mirror images are identical and superimposable, the molecules are called achiral. So for instance, gloves and shoes are chiral. Shot glasses and baseball bats are achiral. When are molecules chiral? If they have at least one or more chiral carbon atoms, a carbon atom bonded to four different groups, or non-superimposable mirror images. So, mirror image of a chiral compound cannot be superimposed on the original compound. When such stereoisomers that are mirrored cannot be superimposed, they are called enantiomers. Here's an example of bromchloriodine iodyl methane. Two mirror images, they cannot be superimposed. So when you rotate a mirror image of an achiral structure, you can align two structures with each other, and their mirror images can be superimposed. For instance, dibromochloromethane and another dibromochloromethane are the same structures because all you need to do to superimpose them is to rotate this image. So in many cases you need to project a molecule that has a 3D shape that exists in 3 dimensions onto a 2-dimensional plane. To do this, we use Fischer projection. So most highly oxidized carbon atom is brought to the top, and vertical lines are used in place of dashes for bonds that go back. Horizontal lines in place of wedges are used for bonds that go forward. So this is an example. So here, in this Fischer projection, okay, these two groups, they go forward. That's a wedge, okay? The dashed bonds project back, okay? In the Fischer projection, vertical lines are used instead of the dashed ones. Horizontal lines are used instead of wedged ones, the ones that look forward. So now we need to understand the concept of D and L isomers. D and L isomers are assigned based on the position of the hydroxyl group on the chiral carbon that is farthest from the carbonyl carbon. The letter L assigned to the structure with a hydroxyl group located on the left. The letter D is assigned to the structure with a hydroxyl group located on the right. So let's take a look. For instance, in L erythrose, the atom that is farthest from the carbonyl is this one. Hydroxyl group is on the left, it's L isomer. In this example, hydroxyl group is on the right, it is a D isomer. Why knowing about enantiomers is important? Turns out that many compounds, including carbohydrates in various biological systems, they have only one active enantiomer. Why does this happen? Turns out some enzymes and cell surface receptors, which carry out metabolic reactions, are chiral themselves. Maybe only one enantiomer interacts with the enzymes or receptors from that chemical. Others are inactive. Or maybe chiral receptor fits the arrangement of the substituents in only one enantiomer. The mirror image does not fit properly. So basically, the proper receptor residues can work only with only one of enantiomers. So for instance, here you have receptor sites on an enzyme surface. This one, this enantiomer, does match the arrangement of the sites. This one doesn't. So here's an example. For instance, we treat Parkinson's disease with a substance called L-DOPA. L-DOPA is converted to dopamine in the brain, which improves the conditions in patients with Parkinson. D-DOPA, which has a different arrangement of the chains, is not effective for the treatment of Parkinson's disease at all. Turns out that for not just for L-DOPA, but for many drugs, only one of their enantiomers is active biologically. For many years, though, drugs that have been produced had mixtures of the enantiomers. Now, since we know that chirality is important, researchers are using chiral technology to separate enantiomers and generate only active enantiomers of the active drugs. So here are some Fischer projections of the most common monosaccharides. Here are the most important monosaccharides: D-glucose, D-galactose, and D-fructose. So just as a reminder that in Fischer projection of carbohydrates, the most oxidized group is always on the top. Chiral carbons are at the intersections of vertical and horizontal lines. H and OH groups are shown on the horizontal intersecting line. And CH2OH group is always placed at the bottom of the Fischer projection because this group is not chiral. So in order to determine whether it's L or D isomer, we need to find the chiral carbon that is the farthest from the carbonyl group. If OH is on the left, it's going to be L isomer. If it's on the right, it is a D isomer. Let's take this molecule. The farthest chiral carbon is number 4. OH is on the left. It is L ribose. If it would be on the right, it would be D ribose. Here you see the same example, the molecule of glucose. When hydroxyl at the farthest chiral carbon is on the left, it's L-glucose. It's on the right, it is D-glucose. Turns out D-glucose is found in various fruits, in the corn syrup and honey. That's, a garden variety glucose. It's an aldohexose, so aldehyde group and 6 carbons with this formula. It is also known as dextrose or blood sugar when it's in the body. It's a critical building block in disaccharides such as sucrose, lactose, maltose, and in polysaccharides such as cellulose and glycogen. If you obtain D-fructose from sucrose, it turns out to be a ketohexose. With the same formula, structurally it's the different one. It is the sweetest carbohydrate, much sweeter than sucrose. So essentially, the sweet taste of honey comes from these two monosaccharides, D-glucose on the left, D-fructose on the right. Galactose. Galactose is an aldohexose that is not found in nature free. It is obtained from the disaccharide lactose. It is a component of lactose. And it has a similar structure to the glucose, except for the fact that at the carbon 4 it has an OH group. In some people, blood levels of galactose become unusually high. In those people, the enzyme that converts galactose to glucose is missing. So glucose is involved in diseases such as diabetes, and normal blood levels of glucose should be around 70 to 90 milligrams per deciliter. So there is so-called glucose tolerance test. You drink special glucose-rich milk, and then a nurse will measure the levels of glucose after you ingest it. The blue line on this graph represents hypoglycemic patient. Yellow line represents patient with the normal glucose metabolism, and red line represents the patient who develops hyperglycemia. What can cause hyperglycemia? Usually it's a diabetes mellitus. In diabetes mellitus, patient's pancreas cannot produce sufficient quantities of the hormone called insulin. That results in the increase of glucose levels in the bodily fluids, and they can go as high as 350 milligrams per deciliter of plasma, which is 4 times higher than normal. There are some symptoms of diabetes which include thirst, excessive urination, increased appetite, and at the same time weight loss. There are 2 types of diabetes. Diabetes type 1 is autoimmune and found mostly in children and stays with them for the rest of their lives. Diabetes type 2 is mostly a lifestyle disease. It affects mostly adults and can be mitigated by the lifestyle changes. In hypoglycemia, the blood glucose level rises and then rapidly decreases to very low levels, as low as 40 milligrams per deciliter, or half of normal. Low blood sugar may result from the excessive production of insulin by the pancreas, and the symptoms of overproduction of insulin may include dizziness, general weakness, and muscle tremors or cramps. The diet for this disease, for hypoglycemia, may include, instead of 3 large meals, several small meals that are high in protein and low in carbohydrates. Now we're going to talk about another way to represent monosaccharides with so-called Haworth structures. Turns out that the most stable forms of pentose and hexose sugars are rings that contain 5 or 6 atoms. These rings are called Haworth structures. They're produced from the reaction between the carbonyl group and the hydroxyl group in the same molecule of monosaccharide. So here we're going to learn how to draw Haworth structures. So first, you need to turn the Fischer projection by 90 degrees Celsius. You have to fold the horizontal carbon chain into a hexagon and bond the oxygen and carbon 5 to the carbonyl group. And then you have to draw the new OH group on the carbon 1 that is positioned below the ring to give the alpha isomer, or above the ring to give the beta isomer. So here's step 1. We're turning Fischer projection in clockwise by 90%. It's D-glucose. Then we fold the molecule clockwise to make a hexagon. and then we bond oxygen on the carbon 5 to carbon 1. And then we need to draw bond between the oxygen of the OH group on carbon 5 to the carbonyl carbon here. And then we need to draw the new OH group on carbon 1 below the ring to produce alpha isomer, or above the ring to generate the beta isomer. So when you place such cyclic structures in the solution, they start opening and closing. Alpha D-glucose will be converted into open chain glucose and then to beta D-glucose. So those isomers change there and back. So essentially, at any time in the solution, D-glucose exists in a alpha and beta isomer forms, and only small amount of open chain is present. So now let's talk about fructose. Fructose is ketohexose. So fructose forms 5-atom ring structure. So carbon 2 will be on the right corner of the ring, carbon 5 will be on the left corner. This Haworth structure forms when the OH group on carbon 5 reacts with carbon 2 in the carbonyl group. You can see alpha D and beta D fructose. So let's talk about some chemical properties of monosaccharides. Sorbitol, xylitol, and mannitol are often used as sweeteners in many sugar-free products because they cannot be used by your cells as the source of energy. So in the solution, monosaccharides have small amounts of the open chain form. It has an aldehyde group with an adjacent hydroxyl group. This aldehyde group with an adjacent hydroxyl group can be oxidized by an oxidizing agent. It can be oxidized to carboxylic acid, such as Benedict's agent. So if you add divalent copper, which is blue, to the D-glucose, copper will oxidize glucose to gluconic acid, resulting in brick red monovalent copper 1 oxide. So sugar acids can be produced from the oxidation of the aldehyde in aldoses, for instance by bivalent copper that is reduced to monovalent copper. The naming comes from replacing the O's ending, the monosaccharide, with onic acid. Sugars that can reduce other substances, such as the open chain form of D-glucose, are called reducing sugars. What about fructose? Fructose is a ketohexose. Ketone groups usually cannot be oxidized, but it can be oxidized in Benedict's solution when and if rearrangement happens between the ketone group on carbon 2 and the hydroxyl group on carbon 1. Such rearranged molecule is then converted to glucose, which produces an aldehyde group with an adjacent hydroxyl, and that can be oxidized. So here's the fructose, which rearranges to form glucose, and only then it can be oxidized in Benedict's reaction. Now what about reduction of monosaccharides? Turns out you can reduce carbonyl groups in monosaccharides, and that will lead to the production of sugar alcohols, also known as alditols. For instance, here D-glucose is reduced by hydrogen in the presence of platinum catalyst to D-glucitol or D-sorbitol. Sugar alcohols named by replacing the O's ending, the monosaccharide, with etol, that can be D-sorbitol, D-xylitol, from D-xylose, D-mannitol from D-mannose. These sugar alcohols can be used as sweeteners in various sugar-free products, for instance, chewing gum and various diet drinks. Now, normally, we're going to talk about the role of glucose in health. Normally, blood glucose is reabsorbed into the bloodstream back in the kidneys. But when the blood levels exceed roughly about 160 milligrams of glucose per deciliter of blood, the kidneys can't reabsorb all of the glucose, and it goes into the urine. And this is the condition known as glucoseuria. One of the symptoms of diabetes mellitus is high level of glucose in the patient's urine, and that can be identified using Benedict's test. Here it shows you mass per volume concentration of glucose, milligrams per deciliter. If there is no glucose, Benedict's test solution is blue. If it's elevated, it turns into blue-green, then yellow, then brick red. Disaccharides. Lactose is one of the a disaccharide that is found in milk and other milk products. It contains two monosaccharides called galactose and glucose. So this is beta-D galactose, this is alpha-D glucose. When they are connected, they form what is called beta-1,4 glycosidic bond. All disaccharides consist of two monosaccharides linked together. When these two monosaccharides combine in a dehydration reaction, if those two disaccharides are glucoses, if those monosaccharides are glucoses, they will form maltose. Glucose and galactose will form lactose. Glucose and fructose will form sucrose. Maltose is also known as malt sugar, composed of two D-glucose molecules. It can be obtained from the breakdown hydrolysis of starch. Maltose is used in preparation of cereals, candies, and brewing of beer, and it is found in both alpha and beta forms. So in maltose, two monosaccharides are linked by an alpha-1,4 glycosidic bond that is formed from the alpha hydroxyl on the carbon 1 of the first glucose and OH on the carbon 4 of the second. So this is alpha maltose. Lactose makes up about 6 to 8% of human milk and about 4 to 5% of cow's milk. Now you can see how alpha lactose is formed. It is formed between beta-D galactose and alpha-D glucose. Now glucose can be both in alpha or beta conformations because that determines this atom. The bond is a beta-1,4 glycosidic bond because OH group on carbon 1 of beta-D galactose. So this hydroxyl group belongs to beta-D galactose, and we always look at the one on the left. It is forming a glycosidic bond with the OH group on carbon 4. So carbon 1 determines whether it's beta disaccharide or alpha disaccharide. Sucrose, also known as table sugar, can be obtained from sugar cane, sugar beets. It is made out of alpha-D glucose and beta-D fructose. And the arrangement is kind of different. So it doesn't have alpha or beta glycosidic bond. Not 1,4. It's 1,2 alpha-beta glycosidic bond between the carbon 1 on the glucose and carbon 2 on the fructose. This disaccharide cannot form an open chain, cannot be oxidized, cannot react with Benedict's reagent, and it is not a reducing sugar. So here you can see how alpha-D glucose forms the bond alpha-beta 1,2 glycosidic bond with a beta-D fructose. This is why alpha-beta 1,2 glycosidic. since we are talking about the sweet things, let's talk about how sweet those things. So various sugars and related artificial sweeteners are different in sweetness. So here in this table you can see the comparison of those sugars and sweeteners compared to glucose. You can see that compared to sucrose, table sugar. You see that fructose is much sweeter. Xylitol, which is often used in the chewing gum, is as sweet as sucrose. But artificial sweeteners that are not carbohydrates can be hundreds and even thousands of times more sweet than sucrose. So here's an example of Sucralose or Splenda. It is made from sucrose, and some of the hydroxyl groups are replaced with chlorine. You can see here and here. Aspartame is also known as NutraSweet or Equal, is a non-carbohydrate sweetener at all, made from aspartic acid and a methyl ester of phenylalanine. Now, one of the important roles of carbohydrates in health is that they determine the blood types. Turns out that the presence or absence of certain terminal saccharides on the antigens, so-called agglutinogens, on the surface of red blood cells determine which blood type we have. If N-acetyl galactosamine is present, it is called type A. If N-acetyl glucosamine is present, it is type B. If none of them is present, it's type O. And if both of these are present in the red blood cell, it's type AB. So O has 3 common terminal monosaccharides. A, as we mentioned, contains the same 3 monosaccharides and 1 extra. B contains same 3 monosaccharides and another extra, while AB consists of the monosaccharides found both in A and in B. So persons with type O blood that contains only 3 common monosaccharides, they make antibodies against blood types A, B, and of course compoundly against AB. So these people are universal donors. Blood types of A, B, and AB can all receive type O blood. On the other hand, type AB blood, which contains all the terminal monosaccharides, they produce no antibodies to type A, B, or O blood, and that makes them universal recipients. So this table summarizes the compatibility of blood groups. You can look at it from your own perspective, what can each blood group receive. Polysaccharides. Cellulose is composed of individual glucose molecules connected by beta-1,4 glycosidic bonds. Polysaccharides are formed when monosaccharides are joined together. Polysaccharides include amylose, amylopectin, and cellulose, which all are polymers of alpha-D-glucose. Amylose is a straight-chain polysaccharide of glucose units. Amylopectin is a branched chain of glucose. So here you can see amylose. Here you can see amylopectin. Amylose consists of glucose units that are connected by alpha-1,4 glycosidic bond, while amylopectin and glycogen consist of glucose units that are connected by alpha-1,4 or alpha-1 and alpha-1,6 glycosidic bond, which plays a role of a linkage, inter-chain linkage. Amylose is found in starch. It makes up about 20% of it, and it consists of 250 to 4,000 alpha-D glucose molecules that are connected by alpha-1,4 glycosidic bonds, forming a continuous chain. Amylose is called a straight-chain polymer, even though the polymers are coiled in a helical fashion, but they're coiled as a single chain. Amylopectin makes up as much as 80% of starch. It's branched chain, and the glucose molecules in amylopectin are connected by alpha-1,4 and alpha-1,6 glycosidic bonds. Starches, amylose and amylopectin, are easily hydrolyzed in water and acid, which results in the production of smaller saccharides called dextrins. And dextrins can be then hydrolyzed further to maltose and finally glucose. Glycogen. It is a polymer of glucose that allows us to store glucose in the liver and in the muscle cells. Glycogen is hydrolyzed in our cells at the rate that allows us to maintain the proper blood level of glucose and allows us to have energy supply between meals. Glycogen is similar to amylopectin, but it is more highly branched. So glucose units in the molecule of glycogen are connected to each other by alpha-1,4 glycosidic bonds, and branches are attached by alpha-1,6 glycosidic bonds that occur every 10 to 15 glucose units. Finally, cellulose, which is the major structural unit of wood and other plants, is a glucose polysaccharide that consists of unbranched chains in which glucose building blocks are connected by beta-1,4 glycosidic bonds. Cellulose cannot form hydrogen bonds with water, which makes it insoluble. Cellulose provides rigid structure to the cell walls in wood and fiber and plants. It is much more resistant to hydrolysis than the starches are, and that makes it indigestible by humans because we cannot break down beta-1,4 glycosidic bonds. So this is cellulose. You can see beta-1,4 glycosidic bonds between individual building blocks of cellulose. And that concludes our conversation about carbohydrates. This is the concept map. I hope you enjoyed this lecture.